Hello, our topics today for video notes is DNA replication, which is from chapter 12.3, and RNA in chapter 13.1. So here's your central question. Can you summarize the events of DNA replication? So moving right into chapter 12.3, we're looking at DNA replication. Think back. What does this word replicate mean? You got it right. It means we are going to copy our DNA. And think about, when does this happen in the cell cycle? So replication is going to happen during our S phase, and we have to copy the DNA so we can provide the new cell that's created a same complete set of DNA. I kind of think of it as like an instruction manual so it can function. So let's look at the details of how DNA replication works. So first of all, DNA must unzip. So we are going to copy this middle section of DNA here. So we must unzip it. And when we unzip it, the next thing that occurs is that there are enzymes that help with this process. So if you look up here, the blue spheres represent DNA polymerase, and we see ASE enzyme, which means it's an enzyme. The enzymes attached to the single strands of unzipped DNA. This DNA polymerase is going to work from the top down, as you can see by the arrow, and this other polymerase worked from the bottom of that separated strand up. And as they attach the single strand of DNA, they're going to add one nucleotide at a time to the new strand. So let's zoom in on this blue sphere right here, that DNA polymerase. And right now it's adding a red piece on, which is a guanine. So the guanine is going to add to its complementary nucleotide with the complementary base pair, which would be G's match with C, so guanine and cytosine. And so the DNA will be replicated by the DNA polymerase until that section is complete. Now, the tops and the bottoms of DNA strands are really challenging to replicate. So there's a different enzyme that helps with that process, which is telomerase. And remember, ASE, it's an enzyme to help speed up that process. So what happens at the top and the bottom of DNA, which we call telomeres, these enzymes will work at those sections that are very hard to replicate to make sure that they add the individual nucleotides at those places.
All right, so there are differences between DNA replication in prokaryotes and in eukaryotes. And so in prokaryotes, remember, think back to our unit in cells when we talked about prokaryotes. Bacteria are examples where they have circular DNA, one long circular strand that connected. And so what happens is they basically just unzip at one side, and they just zip, zip, all the way around until they get to the end of the circle. So that's prokaryotes. And in eukaryotes, the situation with them is their DNA is 100 times larger, and it's linear. And so it's just a long strand. And so what happens is the DNA polymerase has to attach at multiple places within the long strand of DNA so that it can replicate the whole amount correctly with all those little nitrogen bases and letters. They have to match up. So there's multiple places where the polymerase will attach to the long DNA strand in eukaryotes.